Good afternoon, everyone. To all the participants who have joined us on YouTube, I would like to welcome you all to this virtual talk on Agarwood, a potential bioresource for livelihood generation and climate change mitigation in Northeast India. This is a joint venture initiated by the Departments of Human Physiology and Botany of Netaji Subhas Mahavidyalaya, Udaipur, Gomati district, Tripura. Today, we are very lucky to have with us as the chief guest Sri Praveen Agarwal, additional principal conservator of forests, and as the resource person, we are lucky to have Dr. Bimal Debnath, assistant professor in the Department of Forestry and Biodiversity, Tripura University. We also have with us as the patron of today's webinar, Dr. Shudan Debnath, the principal in charge of Netaji Subhash Mahavidyalaya. Acclaimed by several cultures and religions as the wood of the gods or Devya Lakri, Agar wood has gained prominence as one of the most expensive woods to be found. And how did this happen? What is the significance of this wood? We shall delve much deeper into today's webinar. And I would like to welcome you all today as the host to this webinar, which shall be a learning experience and a very interesting journey to discover the potential of agar wood as the economic sustenance for several unemployed youths of our state as well as the world in today's webinar. Therefore, let us now welcome Dr. Deepti Dash, Associate Professor and Head of the Department of Botany, Netaji Subhash Mahavidyalaya, Udaipur, to present the welcome address. Yes, madam.
Good afternoon. Good afternoon and warm welcome to everyone gathered here. It is a glorious moment to extend my warm wishes on behalf of Department of Botany and Human Physiology of Netaji Subhash Mahavidyalaya, Gomati, Tripura. At the outset, I would like to convey my heartfelt gratitude to Dr. Sudhan Debnath, Principal in charge of this college, for allowing us to conduct this webinar. Our chief guest, Sri Praveen Agarwal, sir, Additional Principal Conservator of Forest, Government of Tripura, and invited guest, Dr. Bimal Debnath, Assistant Professor, Department of Forestry and Biodiversity, Tripura University, for accepting the invitation. Before we begin this program, I would also like to express my gratitude to all the faculty members and our beloved students who sincerely committed to this event to make it a success. This event would have not been possible without the support of each and everyone present here. Seminars are organized and conducted for the general audience to gain insight and information dissemination that would remain effective in their life, exercises, and work. The topic for discussion of today's webinar is Agarwood, a potential bioresource for livelihood generation and climate change mitigation in Northeast India. An interesting topic chosen, keeping in mind the interest of everyone. Agartala, the capital of Tripura, is named after the agar tree, which was very common in the area at once. Agarwood tree is internationally known as Aquilaria malacensis. Agarwood is a highly valuable aromatic dark resinous hardwood of Aquilaria species. The formation of agarwood is generally associated with the wounding and fungal infection of the Equilaria trees. The resin is secreted by the trees as defense reaction and deposited around the wounds over the years following the injury, where the accumulation of the volatile compounds eventually forms agar root. This webinar would bring into limelight the traditional, medicinal, and economic importance of the agar plant. Due to its economic importance, Government of Tripura has taken an initiative for large-scale cultivation of the agar root in project mode in the state. Our invited guest and expert, Dr. Bimal Devnath, will be sharing valuable information on the same. I am sure that the program will be beneficial and prolific for everyone present here. Thank you all. Now I would request Patriot Madam to proceed. Over to Patriot.
Thank you very much, madam, for your informative and educative address. Now, may I request and invite Dr. Sudhan Devnath, the principal in charge of Netaji Subhas Mahavidyalaya, Udaipur, to deliver the introductory note. Sir, may I request you to unmute yourself first?
Please. Okay. A very good afternoon, everyone. Thanks, Patriot Madam. Respected Chief Guest, Probin Agarwal Sir, Additional Principal Chief Conservator of Forest, Government of Tripura. Respected Speaker, Dr. Bimal Devnath, Assistant Professor, Department of Forestry and Biodiversity. Secretary Teachers Council, Srimati Patriot Dev Burma, Netaji Subhas Mahavidyalaya. Organizing Secretaries, Srimati Deepti Das, HOD Department of Botany, Dr. Parthasarathy Debra, HOD Department of Human Physiology, Coordinator IQAC, Sri Devashish Rai Chaudhuri, Organizing Committee members, dear colleagues, my dear students, I welcome one and all in this virtual talk. Last 1.5 years, we are facing Corona pandemic and it has changes in almost every aspect of our life. So there is a transition period or transition phase in seminar, workshop, teaching methodologies, etc. from face to mode of virtual mode. In this situation, we have to adopt it. We have no alternative. The main objective of the today's lecture is to aware the students about the potentiality of agar wood for livelihood generation in Tripura as well as Northeast India and how it helps us in protecting the environment. All of we know Tripura is primarily an agrarian state with about 42% of population depending on agriculture and allied activities. However, only about 27% of the land is cultivable, rest being hilly or forest. The climate of the state is suitable for a variety of horticulture plantation crops including pineapple, jackfruit, tea, rubber. A section of indigenous people practices jhum cultivation also. As you know, Tripura is second largest rubber producing state in India nowadays. All of you are familiar and its socio-economic significance. From my childhood, I heard about the story of agar producing plant. The agar producing plant are very costly and expensive. At that time, I thought There may be a fruit like jackfruit or banana, actually, which will be the agar, and which may be agar and which is valuable. Among agar plant, the percentage of agar producing plant are very low. But after that, now I know actually it is not a fruit. The agar plant, normal agar plant, infected by some bacteria or fungus, which creates some resinous product to protect itself. Actually, those infected agarwood are valuable. So initially people were not interested because when the plant will be naturally infected, nobody knows it. Therefore, it was totally uncertain and like a lottery. But in the advancement of science, nowadays scientists discovered the bacteria and fungus which are responsible for infection of agarwood. They discovered it, and now scientists already artificially inoculated the normal agarwood, and this agarwood inoculation convert the price of wood, suppose 5 to 10 rupees to 80,000 to 1 lakh per kg. So that is, non-infected agarwood is like valueless, but infected agarwood, the cost of infected agarwood is nearly 80,000 to 1 lakh per kg. Not only this, from this infected agarwood, if we extract oil, the cost will be likely 20 lakhs. So the climatic condition of our state also suitable for agarwood cultivation, and now government has taken initiative for cultivation and marketing of valuable agarwood. In this seminar, our honorable chief guest and speaker will discuss how much the infected agarwood is valuable and how it will be marketized and how artificial infection will be done. So with these few words, I would like to thanks the organizing committee for choosing such time demanding topics, and thank you all again. Patriot Madam.
Thank you very much, sir, for your highly informative introductory note. Now, today in this webinar, we are truly honored to have with us as the chief guest, Sri Praveen Agarwal, Additional Principal Chief Conservator of Forests, Government of Tripura. Sir is an IFS officer, and today, Sir will shed light and give us a deeper insight into the topic of discussion of this webinar. May I now request and invite Sri Praveen Agarwal to deliver his lecture, sir.
Good evening. Good evening to all. I am audible. Yes, I am asking. Is it audible to
Yes,
all?
sir. Yes, sir.
Okay, that's all. Thank you for inviting Dr. Sudan Devnath and Dr. Bimal Devnath and all the community, academic community of the Tripura who are working in the field of Agar sector. Let me give some brief about the Agar sector and what government is expected to do and what government is doing for development of Agar sector. As Dr. Sudan Devnathji told and about Agar is very valuable species and we know it. In fact, I am also in Tripura since last 25 years. 96, I joined as a professional in Tripura as a IFS professional. Since then, we are also listening that agar is important, very important species. But because of the secrecy around the trade of agarwood and agar-related product, nothing could be moved further. But I'm thankful to the government. They have given me the opportunity since last about 9 months or about a year. We are working in the agar sector, and today after we are in just one position, one position that government of Tripura recently in its last Council of Ministers meeting approved the Tripura Agarwood Policy 2021. I just want to give some highlights and some background how this policy came into existence and how what is the background and what we are there, what is proposed in the our policy. First, as you know, there are a lot of thoughts of going on Agar, how is Agar is going, what should be done, how to ease the rules and regulations regarding the Agar. But they have not a comprehensive roadmap with the government as where we want to go, how we want to go, what is our size of the economy, and how much economy is expected to generate in near future, and how to formalize the entire trade, how to establish the supply chain, backward chain as well as forward linkages, everything. There was no roadmap as such. So from in our policy, we have very much clearly devised the roadmap starting from plantation up to export. Complete value chain we have already covered, and we started working on that directions. Number two, most important point which we understand regarding the other sector, other valuation is, as Dr. Devnath told, we require to have infection of the tree. Earlier, we used to believe only natural infection will take place, but now because of the advancement of science and there are many uh, private providers, private inoculum providers are there who have developed the fungal base or bacteria base, some inoculum, and which if you insert it in the agar tree, then we can get the almost agar root which is nearly very nearly equal to what is we found in the naturally infected agar root. So that when we come to know that technology, then we have also thought we should go for large scale inoculum of inoculum. We should go for large scale inoculum of this other trees. That is one point we have kept in our policy in the mind. Second, as you know, very important, this agarwood species comes under, there is an international treaty called CITES, Convention of Internationally Threatened Wildlife and Fauna, Flora and Fauna Species. And under that annexure 2, this agarwood species is already mentioned. This is mentioned because during 90s, large scale failing was took place about the agar which are already infected or not infected, because at that time people never know if this particular trees are infected or not, and they used to cut it, and after cutting they used to see whether it is infected or not. In that way they have even destroyed the non-infected trees also. Because of that, population of the agar tree reduced substantially, and that enforced the international community to include this species as an endangered species. Now, after that, again plantation started. People understand its importance, and slowly, other plantation also started increasing. As you know, in Tripura itself, there are about 5 million trees are there in private sector, and we want to take the advantage of that. Then after that, we have convinced the government of India. Since we have a lot of plantation, it is not from the wild. We are our plantation are sustainably managed. After every 10-12 years, it is a rotational base. Whenever they cut the plantation, it comes again through coppicing system, or it comes by plantation system. And we should be allowed to legalize this trade, and we should that particular species should be allowed to export. And for that purpose, we have requested the government of India to conduct one study there, we used to call non-detrimental finding study, that is required under CITES. And with our persuasion, government of India hire one scientific authority, that is RFRI Jorat. They have conducted the study, and now they have recommended to government of India for certain quantity of export of agarwood chips and agarwood oil. And once it is being cleared by government of India, then officially we will be our traders will be allowed to export it directly to other countries. And that is another biggest challenge we face. And from last 1 year to 2 years, we and our government has managed to get through that particular obstruction in the formalizing and legalizing the agarwood trade. That second step we have already done, and we are still working, and we are discussing with the government of India to immediately decide that quota for export. That is the second problem, and which we have already addressed. Then problem doesn't stops here. Then we know कि once we start the export, once we start the trading, etc., suddenly there may be chances कि again existing resources may start depleting. In order to take care of that, first point we have mentioned in our policy, we will again supplement the whatever agarwood plantations are there. We will again supplement almost on equal quantity in next 5 years. We are believing that we have 5 million agarwood trees as on date, and we are planning to add 5 million more trees in next 5 years. Maybe in private land, maybe in government land. Main purpose is the population should not be reduced, because otherwise uh, again there's a restrictions from the CITES authority may start. So that is one another addition that we call as augmentation of agarwood trees, that we are planning and we are promoting it. And since agarwood tree is very profitable, we just want to convince the people कि भाई okay, go ahead for agarwood tree. I don't think they require any subsidy or they require any government support for planting. Even uh, we have uh, devised one very good slogan, घर घर अगर हर घर अगर, on that pattern. And this year one more so theme is also on Agar species. So everywhere, if one or two or even 10 trees are there in the one compound of the one person, they may not face any financial problem, and they can, that particular trees can be properly infected through inoculum, they may be in a position to manage the marriages of their children in your futures. That is the beauty of that tree. Even 10 trees you can get about maybe 5 million or and more trees, more cost. And in that way, this is very costly if it is get infected. So we are planning to convince the entire population, all the families of the Tripura, to at least consider planting some agar trees in their backyard, in where whatever space is there. And another important point, we are pursuing the people to at least plant this agar tree as a fencing of their boundary. Once you do the fencing, after 2 3 years, after 4 years, when that tree become a little bit strong enough to become like a pole, then put the barbed wire on that pole, so that automatically infection may start coming to these trees. Because when you put the barbed wire, you have to put the nails, and through nails there may be some nearby bacteria may attack naturally, and automatically fencing of your compound will also be done, as well as it may invite the bacteria to get infected, and it is double benefit, and by the time next 3 4 years it may become infected. So that is also we are planning, and we are convincing entire community to go for large scale plantation of agar trees. Next comes, I already to mention about the plantation. I have already mentioned about the inoculation. I already mentioned about the export. Next question comes about trade, because as on date, people of Tripura, whoever are cultivating and growing the agar tree and making this infection through artificial method, water methods, they are cutting the tree and they are taking it to nearby market in Hojai, which is in Assam, and they are selling theirs, because there is no market, no trade center in Agartala or in Tripura. So another next step we are going to take in our Agarwood policy is to have a one trade center in Tripura, where we are going to invite the buyers from across the country, including abroad, and we are also planning to invite our traders to come here and meet. It is there will be buyer seller meet. First, we will start in a month or time, 1 month or 2 month duration. Then we'll come closer, and then we're planning to have it weekly basis. They should buyer seller meet in Agartala, where people will come from outside and they will purchase the agarwood oil, agarwood chips, etc. directly. And that trade center is very important, and that is also included in our policy. Next question comes, if some traders come from outside, and if they procure some oil from local buyers, they should also know whether the oil which I am procuring is what grade it is, whether it has the same compound which generally other oil used to have, or it is inferior quality or something like that. So for that purpose, they require one testing. So immediately in the same trade center, we are planning to have one testing laboratory also. There, agarwood oil will be tested or agarwood chips will be tested immediately. So before finalizing the purchase, buyer would like to take the test. Okay, they come to the testing center. We will test it. We will do the certificate along with its detailed compounding. What are the compounds available in that particular oil? And based on that, we will decide the quality of the oil, maybe grade 1, grade 2, grade 3, whatever may be the standard quality. And based on that, if the buyer will satisfy, they can take it. They can procure the materials. So that is also one problem we are planning to uh, um, address. The testing is not there at present in our country. Next questions ki I told you about inoculum. As Mr. Devnath told me, there is inoculum available. Now question comes ki bhai, I am the grower. I am having 100 trees. How do I know ki which inoculum I should purchase? From where I should purchase? For that purpose also, it is just like it is just like a vaccine. As you know, the infection creates the antibodies within the body, and what we are doing by inoculum, we are inoculating the trees, and so that the trees start and making antibodies. So now there are multiple types of vaccine like Covishield, Covaxin, etc. In the same way, there are multiple types of inoculum available in the market. So how to know which inoculum is correct or which inoculum is not, because there are many fake inoculum also available in Assam. Many people, they are just making some jaggery, using jaggery some liquid, and they are introducing, I would say that tea becomes some black, but it is not a agarwood. So in order to eliminate such people, we are planning to empanel the proper inoculum supplier along with their rates. We will call for one RFP, request for proposal. Many inoculum provider from national and international community, they may come, they may give us. Then we will take their sample. They will test that in our Tripura conditions, and then after satisfaction and their performance of the agar wood or agar oil after obtaining from that inoculum, then we can empanel them, and then we can say these are three people are there, or these five people are there who are supplying the inoculum. Their rates are this one. Anybody directly can purchase from them, or they can use whatever inoculum they would like to that. So that option we are going to give to the or people or growers in the within the state. In this way, we are trying to approach. We are trying to address the entire entire issues of related to agar sector directly in our policy, and accordingly we are also started working. One important last point is there, as you know, in international export etc cases, we require to have complete uh, supply chain details, from where this particular, suppose one oil is going outside the outside the country, then checking authority at export gate, they may want to know from where this oil comes, from where that tree came, whether it is a tree from legally sourced, number one, or illegally sourced, number two, whether that tree is sustainable managed, or it is just illegally cut, etc. For that purpose also, we need to have complete information, starting from the total tree, that tree, when the tree was felled, when tree was converted, where it is converted, how much oil was removed from that particular tree, then that oil is added into which batch of the oil, and then how it proceed to trade center, from trade center to export on data, export gate, etc. So this complete information will be required, and for that purpose, we are planning to have a big database, mobile base and app base, where every tree will be registered. That registration will be voluntarily, because we are not asking everybody to register. Only those people who are interested to trade, or who are interested to make infection in the agar tree, only those people can register, so that complete data chain can be established, and we can go directly to the root of the tree, where from where that particular drop of the oil came. And such a visionary approach we are going for development of this agar sector, and I request 2 3 points I want to tell about the this academic and scientific community, including students. There are a lot of scope for students, not for livelihood generation. Livelihood generation definitely it is. I already told you every family should plant 10 trees, no problem. But in addition to that, for students, I'm giving this. I want to take this opportunity to inform to all of you for that inoculum, if you develop your own inoculum, if you try for that inoculum, you will that will be one of the biggest patent you can get it, and from that patent you can earn in crore and lakhs rupees. So try to develop your own inoculum within the state using our own resources, and then we can invite some businessmen to start the large scale manufacturing of that particular inoculum which you have developed through using your scientific methods. And if that is can be possible, Tripura can really become, it can change. I can say Agar is the next oil as far as Tripura is concerned. Nobody can stop the development of Tripura if we can develop our inoculum within the state, and that can only be done by the scientific community, like academic community, like you, with whom I am discussing today. That is my first request. And second request, all scientific community may please come to us, please, because we are going in a dark. We don't know anything about how to proceed. Whatever 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 that comes in our mind, we try to address, and we have formulated policy. But still, there are many um, the loose links, and that we have to feel like we don't know what is required in a testing lab, how to go for testing lab. We don't know how to manufacture the inoculum. If some inoculum we can go for transfer of technology. We don't know how to develop the software, because we just imagine कि ऐसा करना चाहिए, but we don't know how to go ahead. So for that purpose, through you and through your faculty, I request if anyone has some idea, they can come to me, they can approach to me, so that we can work together for the development of the state. Thank you very much. That is my inaugural address.
Thank you very much, sir, for sharing your wide expanse of knowledge on agarwood as a potential uh, bioresource for livelihood generation. Today, from your deliberation, from your lecture, we could understand that agarwood may be very potential if it is inoculated either through the natural process or through the artificial inoculation technique, uh, so that it may be, turned into one of the most expensive woods in the world, which is used for preparing materials, or it can be the raw materials for preparing perfumes, essential oils. And no doubt it is because of its use that it has been hailed as the wood of gods. And so we are also grateful because through your deliberation today, we could understand how agar wood may be used as a substantial, in fact, as one of the economic self sufficiency for the students or the unemployed youths who want to uh, establish themselves in a economically. Sir, thank you very much. And we do hope that we would get ideas on how to gain more knowledge and expertise on agarwood
So definitely.
later also, uh, beyond this platform. Sir, thank you very much. So that was Sri Praveen Kumar, the additional Chief
Thank you.
Conservator of Forests, Government of Tripura, who was deliberating in our webinar. Now, I take this opportunity to invite and request the resource person for today, Dr. Bimal Debnath, Assistant Professor in the Department of Forestry and Biodiversity, Tripura University, who shall be deliberating on agarwood as a potential bioresource for livelihood generation as well as for climate change mitigation in Northeast India. Before I give this time to the resource person, I would like to introduce to our participants that Dr. Debnath is an outstanding researcher in forest biodiversity and promotion of livelihood generation in the rural sector of Northeast India for agar wood cultivation and production technology. Sir has done his postgraduate and PhD degree from Tripura University in Life Sciences from 1997 to 2011. Presently, he has been working as an assistant professor in the Department of Forestry and Biodiversity, Tripura University, since the last 10 years. Dr. Dabnath mobilized four research projects financed by DBT, CSIR, and UGC Government of India. He published 42 research articles in various national and international journals. Two students did their PhD degrees under his sensible supervision, and at present, four research scholars are preparing their PhD thesis under his careful guidance in his laboratory. He is working in biodiversity, forest biotechnology, medicinal plants, invasion biology, and forest resource utilization and livelihood generation. Sir is a member and a life member of different societies in India. May I now request Dr. Bimal Debnath to shed light on today's webinar's topic and take this time to give a deep insight into agarwood as a potential bioresource. Welcome you, sir, and the time is yours now.
Thank you, Mrs. Patriot Dabarma. I would like to convey my gratitude to uh, Principal in charge, Dr. Sudan Devnath, patron of this webinar, and today's chief guest, Mr. Praveen Agarwal, Deputy Conservator of Forest, Government of Tripura, and the secretaries, Dr. Dipti Das, Arthur Devbrai. And so I am highly gratitude that you have given a chance to discuss something about the agarwood production and agarwood technology that are using in our state, specifically in the northeastern states. And I also thanks to those members of the committee and the listener of this webinar. Thank you all. Now I would like to share some slides. No.
Dr. Devnath,
Uh-huh.
it is
Yes,
not
sir.
visible.
You may please share your slide again.
Share slide. Slide
Yes,
option
please.
is not coming. Share screen, RI.
Yes, sir. It's the
Share
same.
screen.
Yes, sir.
Is anything visible or no?
Not yet, sir. Yes, now.
Now visible.
Yes, sir. Kindly select the single window that you want to share.
Single window.
The slide window, sir. The middle portion.
So something is there in the slide above the slide.
Yes, sir. Yes, sir. This one. Kindly select it and share, sir. No, sir. Kindly go back, sir. Kindly stop sharing.
Stop sharing. Okay.
Yes, sir. And you can
Here,
try it once again, sir. Please try it once again.
sharing, then share screen
Yes,
is
sir.
coming.
Yes,
Share
sir.
screen. Now which one shall I know? Yes, screen sharing. Share screen.
It has not reached me yet, sir. Could you please share it once again, sir?
Wait, madam.
Yes, sir. Sir has joined us once.
Whether we shall stop our camera? Is there any problem for?
No, sir. Kindly mute your audio. That's all, sir. So, to all the participants who are joining us here on YouTube, I would like to request you all to wait for some time as our resource person, Dr. Bimal Debnath, is experiencing technical issue at the moment, and he shall be joining us very soon. Our resource person is experiencing some technological or internet issues at the moment, and therefore I request you all to kindly join us. Sir will be joining us very soon. Yes, sir.
Share screen. Shall I
Yes,
share
sir.
it? Now
Yes, sir.
after this Windows is not PPT is not showing. They are showing screen sharing. What's again, another dialogue is share screen. Therefore, it is coming. Windows screen share.
So, kindly try to share it once again and then,
Okay, okay. Now it is coming.
yes, sir.
I think it is visible.
Yes, sir.
Okay. Thank you.
Sir, kindly slideshow. Kindly share the slideshow
Oh, slide show.
so that the screen will be maximized.
Now it is maximized.
So, to the top of your screen,
Huh?
shaded in orange color, you will have an option slideshow. Kindly share that.
Now is it visible?
The full slide is not visible, sir.
In my screen, it is in full screen.
Uh, we can see your slide and the slides, other slides also, sir. Kindly select
Now, now,
slideshow, sir. Slideshow, sir. We can see the slides. If you could
hmm.
select slideshow.
Slideshow. I put it.
Uh,
Now,
okay, so please continue. I think it would be fine.
is it visible
So the
clearly or
screen
no?
is visible. Uh,
Huh?
this all the slides and the whole is visible. Whole
Oh,
window is
that's it.
visible. Yes, sir.
Slides are in slideshow mode now.
Okay. It has not reflected here on the screen, sir.
Main slide is visible. Shall I start?
All the slides are visible, sir. Please start, sir. Please
Okay.
continue.
Now see, today we are going to discuss about the agri potential, by agri as a potential by resource for livelihood generation and the climate change mitigation in Northeast India. Actually, whenever we are discussing anything about the agri, then number of questions are coming in our mind. Say everybody else, what is agri? Where shall we get it? Whether this is a fruit or this is something that's are the sometimes scientists are telling these are the some resinous compound. So whether is there any resinous exudation from the tree? So these are the different things coming in our mind. So just to explain these things see here, that is why we want to know what is agar, how is it produced inside the plant, and what is the price of this agorot in international and national markets? Everybody is telling this is the say highly precious wood, and by this by cultivating this agorot say plant, we can earn huge amount of money. Why? What is the price? And what is why it is a precious in international and national market? If it is precious, why it is precious? What it has inside it? And if it is so beneficial, why people are not randomly accessing it? What are the problem? What are the problems of these agro plant cultivations? That these are the some questions that are coming in my mind. Just I want to explain step by step these questions. Now see here. Here, these are the agro plants, plantation, young plantation. Their density is fine. People's planted it. Now, in that cases, you see, this is like a bamboo. Hello, is it visible, Mrs. Petir the Burma? Hello,
Yes, sir. It is visible.
hello. Uh,
Yes, sir.
if
It
any
is
problem
visible.
is there, please tell me. As I am not understanding whether it is visible or not. Not. Just
Sir, it is visible.
only
However, mm,
slide is in front of me. That is in slide sharing mode. So nothing else. I can't see the participant or anything. That's
sir,
not coming.
yes, sir. What you can do is, sir, kindly exit the full screen mode and select a
Uh,
slideshow in your computer. Slideshow. Then the full screen
slide
will have
show.
your slide. Sir, on top of your screen, sir.
Yes. Now it is in out of slideshow.
Doc- Dr.
Yes,
Devnath, in the top, the seventh icon.
yes.
Top. Slideshow. Yeah. Now. Slideshow, which one shall I click under the slideshow mode?
Current
The,
slide, sir. Current slide.
custom
Current slide. Current slide.
current slide. Okay, current slide. From current slide. Okay. Now, is it okay?
Uh,
Same things is coming. Whatever I have given in my past time. Just hello.
yes, sir.
Shall I
It is.
continue?
It is still the same. It is still the same. Or
Same
see,
things.
you can try it from the beginning once again.
Okay.
No, sir. I mean that on that slide itself, you could.
No, sir. Please share.
Hello.
Yes, sir.
Is it visible? Hello?
It is coming. Coming.
Okay.
Coming, sir.
Shall I
Now it
continue?
is.
From the beginning, sir, that the left there is an option on top left from beginning. Slideshow from beginning. Yes, sir.
Slideshow, it is already in slideshow mode.
From the beginning, sir. There. There is
From
an
the
option on the
beginning.
left. Yes, yes.
Yeah, from the beginning. Now?
Sir, I would rather request you to continue your presentation, sir. It's okay.
Okay.
It's not working today.
That means slide is not visible.
Slide is visible, sir,
So what is the
along with
problem?
all the other slides.
Along with other slide.
Yes, sir. So the whole screen is visible.
Whole screen is not
Actually,
visible?
sir, it is in the no,
What
not
happened?
visible. It is in the edit mode.
It is?
In the edited mode, visible but not with the other slides. It is coming.
I am not understanding actually what mode it is coming.
Sir, it's okay, sir. Kindly continue with your presentation, sir.
Okay, so
We
these are
can
the
see your
different.
slides, sir. Hmm.
Okay, these are the different questions that are coming in our explain it. Here you see the plantation. That is, this plantation is very dense. Density is very high. Whenever density, this is the plant actually. This is plant of mixed forest. Plant of mixed rainforest. Specifically, this is the tree of the tropical rainforest zone. So in our state, what happened? You see, that's are getting the suitable environment. That plant grows up to say 40-meter in height, and generally its dBH diameter at best height is 1.5 to 2.5, and this plant support the, see here, this plant support the underground vegetation. That's not disturbing the growth of the other plants. That is why, and it is the evergreen tree. This tree is totally evergreen tree. So that is why it is again beneficial in terms of the climate change benefit. Now you see, this species actually also adapted to a temperature 20 to 22 degrees centigrade and 0 to 1,000 meter above the sea level. The species have adapted in a rocky, sandy, and calcareous soil, oil-drained slope, ridges, and the land near the swamps. And distribute, if I tell about the distribution of the species, is this Acularia gen, under this Acularia genus you see 27 species are reported all over the world under the genus Acularia. In these 27 species under the genus Acularia, 23 species are found in Asian countries, mainly say Bangladesh, Bhutan, Borneo, Cambodia, India, Indonesia, Iran, Laos, Malaysia, Myanmar, Philippines, Singapore, South China, South Africa, Thailand, Vietnam. These are the Agar wood. These are the geographical region where these Acularia species are distributed. 23 total among the 27 species. 23 species are available here, and they have again some of the species are geographically restricted in some areas. You see Malacensis, this is available in Malaysia, Indonesia, Bangladesh, and India. This is India means in our northeastern state. So our say Agaruri link species is Acularia malacensis, and another name is there. This is called Acularia igalocha. And some other species are distributed. Say Krasna. This is distributed in Cambodia, Malaysia, Thailand, and Vietnam. Apiculinia, that is species of Philippines. Equiminata, this is the species of Indonesia, Papua New Guinea, and Philippines. So, like that way, these species are distributed in different countries in Asian continent. And see here, China, they have the Aquileria sinensis. As we have the Aquileria malacensis is our species, and China, their species is Aquileria sinensis. They are commercially use this Aquileria sinensis for the production of the agar root. Now, you see, whenever we are telling about our country, India, so what are the species available under the genus? I am telling Aquileria malacensis. Aquileria malacensis is our suitable agarwood producing genus. Along with these two species are also there. One is Aquileria khasiana, another one is Aquileria macrophylla. Chakravarti 1994 reported that the malacensis is a native to, see here, only the northeastern state, Arunachal Pradesh, Assam, West Bengal, Manipur, Meghalaya, Mizoram, Nagaland, Sikkim, and Tripura. These are this is Aquileria malacensis or Aquileria agalosa, whatever it may be. This is available. This is confined in this northeastern state, and the malacensis species has been reported to occur frequently as homestead of large-scale plantation in the district of Hojai. Everybody knows the term Hojai in Assam, Sibsagar, Sadia, Naga, Derang, Gwalpara, and Kasar district of Assam. That's the area everybody knows. So just for the production of the agar root and agar root oil from this region. And Khasiana is the third species. This is only distributed in our Nicobar Islands. It is not available in the northeastern state. So two species are there, and malacensis is the main agar root. So what is agar root? These are the agar root plant. So what is agar root? Actually, agar root, this is the resin impregnated heartroot of the Aquileria species. Resin impregnated heartroot. So that is highly valuable. Whenever the heart, this would become resin impregnated, then it is highly valuable. Before that, our principals have told us that before that the kilo is 5 rupees. Yes, there is no value. That this is the firewood. That is nothing is usable. And the wood is high demand. Why wood is high demand? Why this is important? So actually, wood is high demand for its some medicinal uses, incense and perfume across the Asia and the Middle East. Now see, as it is distributed in the Asian continent, automatically these are the countries: China, Vietnam, Papua New Guinea, Indonesia, Laos, Malaysia, India, Philippines, Bangladesh, Singapore. These are the countries or agar root producing country in the world. And in India, malacensis is recorded the most agar root producing Aquileria species. And same way, in case of China, they are using, I told it, Aquileria sinensis. Although they have the two species, but Aquileria sinensis, they are commercially. Is there any other plant? Is there any other genus that is used for the agar root production? If so, what is this? Now, you see, another genus is there. Name is Garinops vastegi. Aquileria, along with the Aquileria, Garinops vastegi is the another genus. These malacensis and vastegi are the more popular to the farmer for for the quality agar root production. See, Garinops vastegi. If you cannot see this plant in the fruiting stage, you cannot differentiate. Nobody can differentiate which one is the Garinops vastegi and which one is the Aquileria malacensis. So almost they are morphologically the plants are alike. Leaf structure is almost alike with very little differences there. So only either taxonomist or the scientist can differentiate. The common people cannot differentiate these two species from outside. So here this Indonesia, they are cultivating the malacensis, Garinopsis, and along with malacensis, they are also microcarpa. Microcarpa is another species, Aquileria microcarpa. They are also planting for agar root production. See here. Now, what is the status of this? That is population size of this genus. What is the population size of the genus? So, according to Hasimetal 2016 and Hidayat 2017 and Chaudhary 2016, according to their report, what happened? I have seen that China is the big harbor of these Aquileria species. They have 53,000 hectares with 20 million plants. Almost that means here per hectare, their density per hectare is Hello. Hello.
Yes, sir. Sir, the slide is fixed. Sir, kindly unmute yourself. Sir? Yes, sir. Sir,
Yeah.
sir, your slide is fixed. Your slide is gone, sir. Kindly share it again. Sir, kindly share your slide once again, sir. Yes, it is shared now. Yes, sir. The slide had gone. Yes, sir. Sir, so kindly share it from the current slide. Sir, on your screen there is a button. There is a plus and minus button. Even when you click there, also on I mean to the bottom of your screen, there is a plus and minus sign button. You can click there also.
Hello. Hello.
Sir,
Hello.
sir, please continue with your presentation. When this slide gets stuck, I will inform you, sir.
Okay.
Or sir, you can do it without sharing your screen also. You may refer to your slides and just give a lecture, sir.
Now, hello.
Yes, sir.
Is it coming?
Yes, sir. The slide is coming. However, you will have to, manually use it. I mean, you'll have to change the slides manually.
Yeah, yes,
Otherwise,
I am changing the slide manually. No
okay,
problem.
sir.
But is it
Okay,
visible?
sir. It is visible, sir, but not the full slide. It is not maximized.
Now it is maximized.
Still, it is not maximized, sir.
Till now,
No, sir.
now it is
Sir, please
maximized in
carry on.
my
Please carry on, sir. Yes, sir. Please
in
carry
my
on.
laptop it is maximized.
Okay,
Now
sir.
see slide is moving or not here?
The slide is not
Moving?
moving. No, sir.
No, sir. Is
So,
changing
so please
slide?
carry
No.
on. Please carry on, sir.
It is changing. Slide is changing.
Here on the screen, it's not changing.
In my laptop it is changing.
Okay, sir. Please continue with your deliberation, sir.
If I give it in this mode, is it say, changing? Please see. Now is it
Now it's
changing?
changing, sir. Yes, sir. Now it's changing.
So whenever it is giving a full screen, it is not changing.
When it's in the full screen, sir, you will have to just
Full screen means there is two options. One is here at the base. If you give the full screen slideshow mode, then it will go to slideshow mode. Or on the above the
yes, sir.
say,
It's okay, sir. Please do it as your at your own convenience, sir. No problem. Please continue, sir.
is it
Yes,
moving?
it's changing, sir. Yes, it's changing.
Now you see. mm, I told that the some of the species are distributed. Different species are distributed in the different countries in our Asian continent. So here, I have found that there's a Malacensis. This is I told Indian species, and along with this Malacensis, another two species are there. One is the Khasiana and Macrophylla, but Malacensis or the synonym is another name is Agalusa, whatever it. That is the Indian agro producing species that is used commercially. In case of China, they are using Chinensis. That species is different. That is country to country species is different. I told that except this Aquilarium malacensis, another species is using another species is using. This is the Gyrinopsis Gyrinopsis vastegi. So this species is also used by Papua New Guinea and Indonesia for the production of their commercial agarwood oil and agarwood. Now, I want to tell what is the population size. In terms of population size, you see China cover 5,300 hectares with 10 millions of plant population. But thereafter, after China, India is coming in second position. We have the 20 million plant populations. The density is different in different countries. In case of China, their density is 4,000, almost 4,000 tree per hectare. So this is the density of the plants. This is according to Hasimetal 2016 and Hidayat 2017. Now you see, I want to know whenever I started my work with these plants, just I want to know why this is precious, why it is too costly, what are the reason behind it. Then where this agarwood oil is using. So, just to study these things, whenever I search in different literature, I cannot say I am not understanding why this is too precious till now. See, agarwood oil, this is actually used by many cultures for spiritual, opulent, and aphrodisiac purposes. It is highly revered in the seminal text of Hinduism, Christianity, Buddhism, and Islam. So these are the use. And In compared in Middle East, Asian countries, what happened? They are thinking this agarwood pieces with a high resin content will be bound directly without any processing or addition of other fragrance. In many Middle East societies, agarwood is used as a sign of wealth, status, particularly during the religious rites and social occasions. Agarwood oil, agarwood oil is one of the most five important ingredients. This is another important use. You see, this is one of the most five important ingredients in Chinese perfumery and important elements of Arabian perfumes and prominent in the modern Western fragrance industry. Yes, that's quality say I have seen when I visited Assam. They have given a very small touch of this agarwood oil in my hands, and it was the smell was persist. So more than 24 hours. More than 24 hours. That means some compounds is there that are used as a base of different perfumes, different say fragrance industries. So this is the uses. Another important use is that recent study that was shown that the some compounds are there in this agarwood oil. The one is beta caryophyllene and Ancularia crassiana. What happened? These compounds have anti-cancer activity as a colorectal cancer, breast cancer, and lung cancer. This shows the activity against this cancer. And you see, 367 compounds are reported from these four species. One is Sinensis, Malacensis, Casna, and Saptingera. 367 compounds. Among these compounds are categorized according to their structure, and that's are categorized in different types, 11 types, 11 types. And some major compounds that is the sesquiterpenes, different sesquiterpenes, mixture of sesquiterpenes, and phenylethylene chromes. Phenylethylene chromes. There are 70 sesquiterpenes are reported, and 40 types of phenylethylene chromes are reported from this agarwood oil. And the major component of this agarwood oil, as it is used as a aroma, these are due to the presence of this aromatendrin, beta agarofuran, erythroline, guaiol, and agarosperol. These are the characteristic compound of this agarwood oil. Now the question is how these compounds are producing, or agarwood oils are producing, agarwood oils are forming inside this plant, so inside this wood. Then there are different stories are there, and different articles are there I've studied. Then as a group of scientists, they are telling that whenever there is any wound in nature or by any physical means, then what happens? Some of the microbial flora enter into the plant. They attack the plants. They enter into the plant. So what are the reasons why they will enter only these plants? Only these plants. Then scientists are telling they have some resinous compound inside this plant. That's microbial flora enter into the plant just to feed on this resinous compound. That is why one thing is that whenever they start to act on this compound and they infect the cell, there is two things is working. One thing is that they are cracking the resinous compound into different terpenoids and some other compounds, and side by side plant system is there. Plant system is there. Whenever any microbes are entering into their cell, they are protecting their body. They are protecting their body from this microbe cell, thereby as to resist these, they are synthesizing the different terpenoids compounds, and that as a result is the fragrant compound will be stored in the wood. Then another school there is a by different experiments. This whatever I am telling, these are the natural phenomenon. By artificial way, again there are some procedures that developed that by using these things, farmer can produce the agarwood by artificial means. What happened? What they are doing in case of say and farmers, they are owning that tree. They are owning that tree. That means their target is to facilitate the entry of the microorganism that will produce the agarwood oil inside it. Inside it. So see here. So that means they are by physical or biological. Physical means they are developing their owns, and thereafter what will happen? The biological agent will enter into the system, plant system, and that will enhance the development of this agarwood production. And this is because this agarwood, artificial agarwood by the fungal elicitor show the similar oleoresin. That means some experiment were done. That means whenever this fungal elicitor are entering and entering into the in the healthy plant, then what happened? That is produced. They are inducing the production of some oleoresin compound that is present in the agarwood oil. However, agarwood is formed only after the trunk or branches is owned or infected with the microbes. This is actually the stress. This is they develop a stress which induce the defense response and change a secondary metabolite network, resulting in the accumulation of the sesquiterpenes and phenyl ethyl chromes derivatives and the deposition of the resin in the tree. They are scientists are telling these things. Since sesquiterpenes and phenyl ethyl chromes derivatives are the main compounds in the agarwood, and understanding the biosynthesis of regulation of sesquiterpene and the chrome in Aquileia species is critically important in determining the mechanism of agarwood formation. And sesquiterpenes belong to a large group of metabolite family. This is the terpenoids. The chemistry people knows it better than me. And all terpenoids, these are derived from the common precursor, that is isopentenyl diphosphate. Which is synthesized by two different pathways. One is the MDA pathway, another is the MA pathway. That is also chemistry. In case of isopentenyl diphosphate, where adapted to condensation reaction to generate a linear polymer, and in the final step, what happens? These terpenes are formed by terpene synthesis gene and terpene synthesis from the linear polymers. Now, studies on this induction mechanism of the terpenoid biosynthesis of agarwood showed that the methyl treatment increases the expression of terpenoid synthesis gene that is activated by methyl jasmonate and induces the production of this sesquiterpene delta goins in the cell culture. That's experiment is done in plant tissue culture in the laboratory. They have taken the tissue of Aquileia khasiana and Aquileia sinensis. So I told that sinensis, this is the species, the Chinese species, they are using it. Another interesting phenomenon is noted. What is this? The pruning activity growing that assemblings of Aquileia sinensis resulted in hydrogen peroxide burst. This exogenous hydrogen peroxide could induce Aquileia sinensis plant to form the vessel occlusion. That means vessels will be closed, vessel occlusion, and produces the sesquiterpenes. Very interesting. Then based on these experiments, actually the chemical inducers are developed by the scientists. Based on these experiments, say methyl jasmonate, hydrogen peroxide treatment. These are the different artificial method for the induction of the sesquiterpene synthesis in plants. These are based on this experiment. Further study in culture cell suspension. Culture cell suspension of Aquileia sinensis found that hydrogen peroxide could promote the program cell death and salicylic acid accumulation, sesquiterpene production. See here, suspension culture of the cell. Whenever hydrogen peroxide is given, then what happens? Sesquiterpenes is producing. So this is indicating that the hydrogen peroxide also is sensitized to the TPH gene. On the other hand, by prosequencing the small rRNA subunit of non-contaminated own sample and sample open to the environment, non-substantial variations was observed either in the fungal or bacterial enrichment, and diversity, or in the relative abundance of the taxa. This demonstrates that only in the absence of brilliancy here. These experiments are demonstrating that in absence of microorganism, only in absence of microorganism can induce the plant for the synthesis of these sesquiterpenes. That is microbial independent technique. Without microbes, plant can be synthesized these sesquiterpenes. That is the agar root oil major components of agar root oil. Now, in case of microbial infection, what happened? Up to 2019, it was found that the total of 340, a total 340 fungi and 131 bacteria were isolated from agarwood, stem, agarwood plant, infected agarwood plant stem. Along with this, what happened? The scientists are trying to correlate where these microbes are coming. The 340 microbial fungi, that microbial flora, 340 fungi, 131 bacteria. It is a very huge number of microorganisms. Whether all microorganisms responsible, or some microorganisms are responsible, some are not responsible for the synthesis of these sesquiterpenes or some other components of this agarwood oil. To study this thing, what happened? The scientists again tried to correlate. What are the microorganisms play the major role? Then in that cases, what happened? 113 species of agarwood endophytic fungus was identified. They are somehow correlated with the production of the agarwood oil in agar. And another group of scientists, they are telling that 31% of bacteria and 23% of fungal isolate 23% of fungal isolate only showed their ability to production of the agar-spiral. That means they, by infecting it, they try to find what are the major components they induce the synthesis inside the cell. They found that they are initiating the synthesis of agar-spiral by artificial infection methods. Now we'll see. These are very big flora. I told that the fungal flora is very big, connected with this agar-root formation. In that cases, Fusarium species shows maximum diversity. Fusarium species shows maximum diversity among all the fungus. A huge number of the Fusarium species are found along with this agar-root, infected agar-root. Now you see the fungus flora that's reported 78%. Role of these fungus, that is the 78% of these fungus are not till now investigated. 78% till now not investigated, and only 8% associated with the agar-root formation. How many microbes are reported? And the fungi not associated, that is 14% fungus were found that are not associated with this agar-root formation. Actually, whenever agar-root is formed inside the stem, then these fungus are attacking, and then these fungus are entering into the system. Now you see, again, among these microbial flora, they are again distributed geographically. Their geographical distribution is different. You see, in this diagram, say different species of fungus are there. Is there any similarity in Indian? See, these are the Indian seven species are reported that are directly connected with the agar-root formation, and 4 species are from say China. Is there any similarities? Indian species, Chinese species, Indonesia and Malaysia? Yes, only two samples here, 2 countries, China and Indonesia. They shows a common microorganism. They shows a common microorganism only that can induce the biosynthesis of the terpenoids inside the stem. Now you see, in case of bacteria, I told that the huge number of bacteria are there. 8 bacteria are again reported by both. He's claiming this 18 bacterial culture that is culturable. See here, whenever we are telling the microbial flora and the whether it is the virus, a bacteria, or a fungus, then there is a question whether these are culturable, whether these are culturable. If it is culturable, then what are the say media? What is the media? What is their protocol that we can develop their culture? So that everything have to say standardize. Whenever that's to standardize, only these culture, these protocols can be used. See here, one in this case, I would like to still see these are the species: Xylaria, Lecidophoria, and Colatotrichum, Botryosporia. These are the species, promising fungal isolate, which deserve to the scale of the commercial level of the production of agar oil, and chemically stimulated with formic acid, formic acid, and after formic acid, chemical stimulation with the formic acid, then introduction of the Botryosporia, Doetheria produces the high yield and the high quality of artificial agar in relatively short time. So that's reported. I am telling you, see what say in case of culture, you see in case of culture medium, that culture medium have to say finalize where these microbes can be culturable. Otherwise, we cannot produce this culture. In that cases, I want to cite an example there. You see, if I tell about the mushroom, now the young youth, they are producing the mushroom at their houses and selling in the market. But see here, in that cases, they are producing only that mushroom, that fungus, that mushroom, which are already for which protocol is standardized, protocol is available. By using this protocol, they can they are cultivating it, selling it, earning something. But if I tell about some important, highly precious, price is very high. Principles of no see, say 1 kilogram, say 800 rupees and 1,000 rupees. This is the mushroom. This is that mushrooms are growing on termites nest. So this is important mushroom, but till now there is no protocol for the cultivation of this mushroom inside the laboratory. If this protocol is developing, if we can develop this protocol, then what will happen? See here, people can earn huge amount of money. Its demand is very high in the market, 1,000 rupees per kilo. Can you imagine it? Yeah, can you imagine it? So see here, so this there is so that is why in everything, every cases, if it is the macro, then we have to standardize, we have to identify. First time the media selection, identification. These are the some important criteria. See here, these are the agar pieces that I collected by using an instrument from the trunk of the tree. For the collection of the presently, it is not needed to fell a tree just to test whether the agarwood is say developed inside it. We have the instrument. You can collect a very small pieces of wood from the stem, and thereafter you can test whether the microbes are there. I have tested it. See here, no. We keep it a long time in a incubator, but there is no development of any microbial flora from this wood. Now you see, whatever I am telling, say these are actually that's what we have done in our laboratory. That it is the media selection is important. I told that, and that's a different media are selected. So for this media selection, hundreds for a single sample, hundreds of culture we have to develop. After this development, we have to identify it. For this identification, what happened? We need again the molecular identification. Without this, nobody can tell by only a mycelium, observing this mycelium, what is this? How can you tell? You have to identify by molecular characterization. For this molecular characterization, see here, molecular characterization, you see here. These are molecular collect. For this molecular characterization, we have to collect its 16s. We have to isolate its 16s RNA gene sequence. Thereafter, by gene sequencing, you can identify this. So this is another job for identification of this. Now, these are about this the natural phenomenon, natural way of inoculation. Now, see here some other phenomenon that's are related to how our farmers are using. See how our farmers are infecting this, how our farmers are infecting these plants for say artificial inoculation. They are say putting the nail throughout the stem. You see, and they are developing some wounds, developing some wounds. Actually, their target is to enhance the natural agent to enter into the plant body system. This technique, this is a common technique. Everywhere farmers are using this technique. That's picture I collected from our state Dharmanagar. Now see, I told that some places are restricted. I say in case of Tripura, only north district, not Dharmanagar. This is from the north district is say prominent, and their farmers can cultivate, can produce the agar root, but in other side it is not producing. So what are the reason? What are the reason? And another thing is, if I tell about the Assam, Hosa is more dominating. And although Shivsagar, Golaghat, and some other Jorhat district, they are also producing. So why in all the places that are not producing? There is one thing. So whether soil is different or the microbial flora that are inducing, that is absent in some places. Just to study these things, we have collected the sample from the different geographical position, different corner of our state. This plant I have collected from South Tripura. Now, if you seriously observe, you take and observe it. Just you observe it. See here. I would like to make it. Is it visible now? Hello? Hello?
Yes, sir, it is visible.
Now you see some of the holes are there in this tree. But small holes are there on this tree throughout the body. Just you see here where my cursor is there. You see some holes are there. Holes are there. So what happened? Who did it? What else? Whenever say inside this tree, what happened? Say agarwood formation is started, then that type of holes will be available. What happened? An insect enter into this. An insect caterpillar enter into this trunk. They are doing this hole, and they are entering either to feed on, or whenever they are doing this, they also enhance the entry of the microorganism through this hole. Now see, this is a old hole. This is old hole. This is the hole. That means caterpillar enter into this. What is this? Now see, this is the insect that's are actually feeding on this stem and entering into this stem. The name of this insect is Geozera confarta. Geozera confarta. That's are entering into the, after that means they are enhancing the that is natural inoculation. They are helping in natural inoculation. That means whenever this insect are entering, then farmer understand that okay, this plant will give us fruit. That means we will get something from this plant. See here, say if you do not see this insect, but another symptoms you can observe. You see when the peoples are visiting their forest, how they will understand whether the tree is ready for felling. See here, under the tree, these are the fecal matter of the insect, Geozera confarta, and thereafter you see huge amount of fecal matters are there. Then they understand, yes, the tree is infected, necessarily infected. Now they will get this. Now we see for this natural, I told that there is one is the artificial method, and other natural factors they are enhancing the production. If it is the artificial method, it has some natural factors are inducing this production, induction of this agar root formation, then there is some advantages. The quality of agar root is very high in that cases, but it takes long time. Little long time, it takes long time. So this is their weakness. So like that way, whenever we are telling say conventional method, that's a farmer using, again that has some merits and demerits. You see, say whenever they are using conventional method of induction, then what happen? They have to use the labor. Their labor cost is say increasing. That means ultimately what will happen? Their production cost will be maximum. Production cost will be the maximum. And in a non-conventional method, that is the biological method. This is non-conventional method that's are available in the nature. Now in the literature I have seen different agar root kits are available. They are telling one is Agar, and another is the agar root with kit. Then these kits are available. These kits can be used. That means they contain some microbial flora. When farmer will inject it, then it will enhance the agar root formation. Agar root formation. Now, see, in case of chemical inducer, I told that how this chemical inducer they decided. See, hydrogen peroxide is there, formic acid is there, along with this hydrogen peroxide and formic acid, they are again using the agar and agar and CAK. These are actually the material. That means they are enhancing by using these chemicals. They are enhancing the infection of the infection of the plants. And now you see in chemical that's they are using, one is the I told that jasmonate and methyl jasmonate, soybean oil, brown sugar, sodium chloride, formic acid, hydrogen peroxide, salicylic acid. These have been used for stimulating the agar root formation. This was reported by Chan et al. In 2011. Now see here. The plant, I told that the Geogyra conferta, that's enhancing the infection. See here, when these insects are entering, these insects are developing some board. They are boarding this trunk. Then what happened? Then the agar root form in these plants is, in terms of quality, they are more better. They are more better. That means insect wood infested wood is that is more valuable. The quality of agar wood is very high. So these are the some plantation we did our work in Dharmanagar district, and this is the picture. See here, that is from Assam. This is from Shivsagar district, and how they develop the cottage industry like. That means these people are working. What is their duty? They are isolating the black wood. Actually, whenever infection is going on, then what happens? The wood become black in color. Black in color. Then they are cutting the tree, and thereafter they remove all the white wood, soft wood. The wood is very soft, they easily can remove. So they remove the white wood and only the black wood portion is separating. After separating, they will give to the say owner, and they will give 700 rupees per 3,000 rupees per kilo. 3,000 rupees per kilo black wood separation from this wood. This is the rate. Sorry. Now see, these are the black wood. These are the black wood. They are depositing the black wood. Thereafter they will measure it, and then they will get the 3,000 rupees per kilo black wood separation. Now see here, these are the infection is not good in their cases, but they are trying to collect. They are trying to collect black wood from here also. See here, after removing the white portion, they are depositing this black, and like that way they deposit all the seeds. They say black agar root seeds in these bags. Thereafter, they are using these seeds in sending these seeds to their fermentation system. That is water hydrodistillation system, and their hydrodistillation system they develop by themselves. This is the area in the Shivsagar district. I visited this area, say before the coronavirus, say COVID started. So in that cases, what happened? From the national highway, Guwahati to Zorat national highway, I entered 25 kilometers, 25 kilometers, and I saw that most of the home gardens, they have a very big plantation, very big plantation, and everybody they planted some of the plants. I told that whenever we are extracting wood, we are extracting the say wood pieces from the healthy plant. We can test whether there is any agarwood formation started or not by just collecting a piece of wood, placing it in calcium medium. So I see here these are the non-infected wood. See here completely there is no infection, and here the black wood that's necessarily infected. And I just told you this. These are the different microbes actually we identified. Whenever we are isolating these microbes, we had to say isolate these. We have to characterize this. Characterize, that means first is the morphological characterization, therefore is the chemical characterization, later on is the molecular characterization. Thereafter we can tell, oh, these are these microbes. And so these are the different isolate we isolated. I told that this is the molecular characterization. Without molecular characterization, nobody will believe us. So this is the condition whenever we try to develop our consortium, and we introduce it, but after 3 months we get this result. But ultimately we cannot claim yes our consortium is working, because we have to test this wood, whether there is our microbes are there or some other microbes from nature are infected by this way this is produced. So these are in this stage, already in the research stage. These are the research stage till now. See here, these are artificially induced agar, like that. That is where you induces only that's agar formation is localized in that area. That is not spreading throughout the tree. But whenever you are inducing some chemicals, then that's distributing throughout the tree. See here, this is a naturally infected agar. This is naturally infected agar, and this its quality is very high, very high. Its quality is very high, and density. Whenever it is become black and it is impregnated, then it is found that density is increasing. It become somewhat heavier than the normal wood, and if you keep it in water, it will mass. So that is by this way they are testing whether this is the impregnated agar wood or some black wood is given, or decaying wood is given. If it is the decaying wood, it will not it will float on the water. Whenever it will float on the water, then there is no synthesis of this oleoresin in this agar wood. Now you see, whenever they are working, just I talk with them to know what are the say how they are earning, how they are getting money by doing these things. Just they share their say livelihood process with me. And I told it, now the price. Oh, the price. The price is different. This blackwood price is different, and agarwood oil price is different depending on their say quality. Quality is here plays the maximum role. Say that means it will, by the quality, its price will be fixed. So in our Dharmanagar market, I have seen that these blackwoods they are selling. They are selling in 80,000 to 1 lakh rupees, and if quality is very high, then 1 lakh 20 rupees per kilo wood. See here, in that case, whenever it is totally a black wood, they are not extracting oil from this wood. They are selling wood directly, and these woods are taken by the some businessmen or some group of peoples. They make it dust, and they are selling this wood dust in the form of wood dust. Thereafter, they are processing it in different way. So they are not farmers are not extracting the oil. They are disinterested to extract the oil. If they are extracting oil, then its price will reduce. Because their oil quality is that they are using their own system, own hydrodistillation system. I shall show how they are using their own hydrodistillation system, and that is why the quality automatically the quality of this oil will be the somewhat lower. Now see, thus in case of Singapore, I told that Agartala, sorry, Dharmanagar market, but in case of Singapore, they are selling this 5,000 to 10,000 per kilogram, $5,000 to say $10,000. So this is very costly. $5,000 is very high, depending on the quality of this, and it contributes significantly to the if price is like that, so automatically it can contribute the significantly in our economy of the local people. So that is why it is so much interesting and important in respect of if you can in respect of livelihood generation in our say youth peoples. Now you see the different times, different news are coming. This is agro-chip market. They are telling that in 2019 to 2029, they are that means economy that will say generate from this agro only $64 billion. $64 billion. So now you can imagine what is the market in our say head. So in future, what is the future market? Future market is very high. That's telling by different say newspapers. Now see Assam, what happened in case of Assam? The farmer industry gets the boost as government and say agro-chip crash. That is come in the Asian age. That is Asian age. So that means Assam government also took some initiative for its development. Although government are taking initiative, they are telling, okay, you cultivate it and no problem. But the plant is under the category, that is critically endangered category. It is critical endangered. This plant, both the species, Aguilera melacensis as well as the Garinopsis, both are under this critically endangered taxa. This is the critically endangered taxa. And melacensis, Aguilera melacensis, it is now already extinct from the wild state. It is not there. So in our forest, whatever we are seeing, actually that's are planted by the Department of Forest, government of Tripura. And home gardens, whatever we are we have seen some photograph, that's actually developed by the farmers develop these things. This is not the natural plantation. So it is already extinct from the wild. That is why according to this, as it is critically endangered category, so its business with this species is completely prohibited. Actually, in this case, I want to share my opinion on that here. See here, my opinion is that these plants are, if I tell about the rubber plant, have you seen any rubber plant in the forest? I think no. No rubber plant is in the forest. But it is it threatened taxa? No. In the wild state, it is not there, but it is in cultivation state. It already rubber can occupy the maximum land in our state. But it is cultivated. It is profitable business. That is in 1980 decades, I have seen government of Tripura, they give some intensive to the people to make the to develop the plantation. But now they are not giving any intensity. They are telling no, not necessary to increase this rubber plantation. But people are not listening. They are developing it by their own interest. Why their own interest? Because the economy of the state totally led by this rubber cultivation and rubber cultivation. Rural economy is totally based on this rubber cultivation. So that is why they are practicing it. Nobody have to tell it. If you declare, if in that cases, see here, it is restricted. Its market is actually not the open market. Till now, its market is not open. So it is a closed market. Closed market system is going on. And if we can declare, I do not know whether is it possible or not. If we can declare this plant as a plantation crop, then what happens? Nobody will collect this plant from the wild state. Everybody will start to cultivate it for their own interest, own interest. Whenever people will cultivate, and automatically different businessmen, different say company, they will come here. See here, rubber to sell the rubber sheet. People are not going to Delhi or Mumbai. They are selling it here. The people are going to plant pharma, say whether it is available. So like that way, so that will depend on the interest of the farmer, interest of the local people. Whether local peoples are more interested, then that's can easily plantation will develop, and however plantation will develop, so it will not be in extinct state. This population size will increase, automatically population size will increase, and people will plant more and more. I have seen in Dharmanagar they have a plantation, side by side they are also increasing. Hello. Mm. So this is the situation. So I told about the Ayush Centric. Government of India in 2014 they have developed a draft policy for the sustainable irrigation of Agar road. At that time, scientific community requested them to make it say free. That is, Aguilera species, Aguilera meleagrensis free from this Ayush endangered category. So now government are taking some initiative for these things. So this is the Assam Gazette. They have notified some objective of this Agarut, sustainable development of the Agarut. Government of Tripura is also there. They took some. They are the two scientists. One from Pune, Agar Pune, Dr. Khaddu, and another is this scientist from Gomati University, is the biotechnologist and molecular biologist. And she is also working in our group. I visited the Assam along with them. And thanks for this, for listening me and giving me a scope to deliver something about deliver a speech on something about this agro tree and how we can use it for the development of the livelihood. And if you want to see how they are processing, one video is there. So I think I took more times. I would like to stop here. Thank you all. Thank you all. Now, uh, Madam, hello.
Yes, sir. Thank you very much, sir, for a very wonderful and informative lecture on how to consider agar wood as a very potential bioresource for livelihood generation, as well as for climate change mitigation in Northeast India, especially a place like Tripura. So
Hello.
we have several comments on the chat box which give witness to the fact that the lecture was very, very informative, and all the participants have been truly enlightened by your lecture. They are also raising several questions and queries in the chat box in YouTube, and I shall try to take up the questions which
Okay.
have been
Okay.
put forward in the YouTube. So, first of all, I would like to take up the questions which have been put forward for Praveen sir.
Uh-huh. Yeah.
So, let me take one question at a time.
Dana.
Question is.
Our chief guest,
Uh,
who is the additional principal
uh,
conservator, chief conservator of forests, has
uh,
not been able to be present till the end of the meeting due to an urgent meeting which he has. However, he has tried to answer the questions which have been raised by the participants. And as to the first question that Krishnendu Das, assistant professor in the Department of Maths NSM, had raised about secrecy in trade of agar, sir has said that everyone knows that agar wood is costly, but no one knows how to extract it. So, uh, No one knows how to trade it, where the trade demand is, etc. Now, this is because those who are involved in trade keep it a secret, and still it is very secretive.
Yes.
I don't know how far this question has been answered, but sir, would you like to put your own observations on this question?
Yes. What happens? So it's a extraction procedure that I have a video. I can show these things, no problem. But extraction procedures, that is, those are cultivating. They develop their own extraction procedure. This is just a hydrodistillation process. They are immersing these ships in water for few days. Whenever they will soak huge amount of water, thereafter they will keep it in a vessel. Then they will heat it. The oil, that is the oil part of these wood then comes with water through a pipe, and they are circulating this pipe through a water tank. What will happen? That will cool again. It comes, the oil condensed and comes with water, and they are storing it. Thereafter, oil that's floating on the surface of water, they are separating it. This is the technique they are using. This is not a problem. Problem is the market. I told that market is not, till now it is not the open market. This is the problem. That is why people are not interested. If it become the open, nobody have to tell you cultivate it, you do it. Everybody will do it. See here, if I compare it with the rubber plantation, nobody is telling you develop your rubber plantation. People are doing it. People are more interesting to do it. So same way, whenever it become free, no problem. I can travel with this wood. I can sell this oil in the market. So I can carry this oil, I can carry this wood. No problem is there. Then automatically business will develop, and small scale industry will develop, and people will come in there. See here, Huzai already small scale industries are developed. People are most of the people, they have the distillation unit. Most of the people are cultivating it, they are processing it. This is their common business. That type of business, if we want to develop here, we have to make it free. Nobody will interfere. The farmer will carry this wood, farmer will carry oil, whatever they are doing. Thus they have to restrict. They do not interfere in the government plantation forest. They should not collect anything from the forest, but they can, whenever they are developing plantation, they can use it. In Dharmanagar, when I first time visited, then they are asking me, sir, what we did? I have developed a plantation with 5,000 plant. I cannot sell it. I cannot collect the wood. I cannot sell it in the market. I have to sell everything in say underground market. I am not getting the actual price due to this. So this is their, they told me these things. They shared me what is their problems. This is the problem. So that is why we have to make it free. As the cultivation, say plantation crop, like a plantation crop, tea, say rubber, then some other crops that we are developing in plantation. Thank you,
Thank you very
madam.
much, sir. Uh,
Then,
to another question which was raised by Roger Das. As these woods are expensive, so it might need extra cost to grow and develop this wood. How can one start up these? To that question, Praveen sir says, no, plantation cost is just like other tree, maybe around rupees 100 per tree. What is costly is to inoculate the mature tree and may cost anything between 2,000 to
uh,
10,000 per tree. That
here I want to
inoculum
add.
will make, yes, sir. So that inoculum will make the normal agar tree into the agar wood.
So
Therefore, the question is very already
in the first
answered,
question,
I believe.
in the first question, I will tell you that
Yes.
yes, plantation is not costly. But with which species, which variety I shall develop my plantation? This is the questionable. See here, some of the species I have seen in Assam. What happened? The plants say diameter is hardly 500 centimeter. That means 0.5 meter. They are felling it. I asked them, what happened? Why you are felling it? Well, they are telling, sir, it is already ready. We will get the oil from this plant. They fell it. And in our state, what happened? Some of the plants that's a species that are using in Dharmanagar, they are commonly say missile infection is more in terms of the south district. In case of South District, I showed one very big tree, not one here, there is some other tree also. Very big two trees are there. In that trees, nasal infection is there. So that means we have to find some reason behind it before giving to the farmer. See here, farmers they are laymen. They do not know anything. Whenever we will tell, they will cultivate it. But we have to sure that farmer will get some benefit from that species. So this is the one thing. That means we have to confirm whether the genetic variation is there. In case of MBB college, more than 60 plant or more than 60 plants may be there. I have collected some wood from this plant to test whether the infection is there. I have seen no infection is there till now. Age most probably the age of this tree is more than 40 years, more than 40 years or near 50 years. So we have to find this reason. What are the problems? Or otherwise, we have to collect the breed from that regions and to distribute the farmer. Yes, you cultivate it.
Thank you, sir. There is another question which was also put forward to Praveen sir, and the question is up here on the screen. Since this place had a sizable number of trees up to a time, the local biodiversity must have had an innate relation with this tree. Is there any such study taken up by the forest department so far? And to this, Praveen sir has said that no, we do not have any study. Moreover, all these trees are in private land and not promoted by government. People on their own planted these trees. And there is also a follow-up of this question given by Dr. Nupur Datta madam, who has also said that that this okay.
What is the question, Nupur Datta madam?
Yes, the question is here on the screen, sir. And
Jai Krishnandu Das, Nupur Datta madam.
yes,
Where is it?
on the screen, sir.
Please,
Not-
can you read it?
Yes,
Here only Krishnandu, question from Krishnandu is coming here.
yes. The question is given, and the answer has also been given by Praveen sir. And there is also an additional information given by Dr. Nupur Datta on the chat box, which I would like to share. Okay. That is, as far as I know, Agar wood is not protected by Indian Wildlife Protection Act 1972. However, it is protected by state bans under Indian Forest Act. Sir, would you like to give any
Yes, under
extra
Indian Forest Act.
information?
But so in that cases, we cannot do anything. So government of Environment and Forest, government of India, they have to take the decision. That is why problem is here. Even state government can allow the people to cultivate it, but they cannot change the rule. Yeah. If anything have to declare, say, bamboo is declared as grass by the government of India, Department of Environment and Science. So this work have to do by the government of India. State government cannot do it. Just relax. State government can relax only.
So there is another question. Do the saplings generated from trees which are easily infected or at least bear infections more likely to be easily infected in due course? Any study on that, sir?
No, in
This
case
has
of
been
yes,
put forward by
once
Krishnendu.
in our group, one scientist is working from JNU, Jawaharlal Nehru University. Now the work has actually ceased due to COVID situation. We had started at once. So in that cases, he tried to make some infection in seedling stage before distribution of the seedling, but it was unsuccessful. That means plants do not receive these microbes. Either those plants are dying or they are protecting it. They are protecting it. They are the cell where infection was done. This region is separated from the other part of the plant body. This is the normal phenomenon.
Perhaps this has also been answered during your deliberation. However, sir, I would like to put it again. Which yields better, natural inoculation or artificial, sir?
Natural inoculation is better. Natural inoculation is better in comparison to artificial. What happened? If artificial, if we say induce the natural inoculation by and enhance the natural inoculation by the farmer, this is actually same thing should be happen. Whatever we will get from artificial natural inoculation, the wood quality would be the same.
Okay. However, we can find that natural agarwood trees are very rare now. They are very rarely
Tree is
cultivated.
rare in wild state, not in home garden plantation. This is available, but it is rare in wild state.
Yes, sir.
Wild state.
And there is another question. Are the saplings of agar available in Tripura?
Yeah, it is available in specifically Department of Forest. They are producing huge amount of seedling. More than 1 lakh seedlings are available in South Tripura Peratia Forest House. One nursery is there. I have seen in the last year more than 1 lakh saplings are there. They are selling it. They are selling it. Price most probably at once it was only 2 rupees. But recently, I cannot tell what is the price. That was only
Okay,
2
sir.
rupees. That means we have to pay for 2 rupees per sapling.
Okay, sir.
And it is also available in Dharmanagar district. They are
Okay.
private farms or the farmers they are producing every year. So, if anybody want to collect the sapling from that area, so we have to tell them before. Just now, if we tell now, they can produce the sapling. Now is the actual season. Now the seedling that is fruit and seed shedding will start in this month. That is in the July.
So it is a great news for all those
Now
aspirants
they
of.
can everybody can develop plantation now. This is the season.
Yes, yes.
Already
Agarwood plantation
seed
that
shedding started.
saplings are easily available.
Yeah, saplings are easily available.
Okay. And so, what are the weather and soil
Weather condition
conditions?
generally it likes 20 to 22 to 30 degree. This is their favorable temperature, but they are surviving in Tripura, Assam, very suitable. This environment is very suitable for this species, and it is not available in higher region of the Meghalaya. Just say Shillong or some. That is altitude, higher altitude is very high. This species is not there. But in other all the conditions that's are growing very nicely.
And sir, what about the conditions, weather and soil conditions for the bacteria and fungi growing? I mean, for the inoculation, perhaps the
Inoculation
person
actually
does wants to know.
inoculation means different scientists in different places they are trying to develop. So different specifically Chinese they develop different kits. So in India whether any kits are available do not I do not
And
know.
the
And
maturity of the
and
woods, sir, for
uh,
the right time to get infected, what would be the maturity of the woods?
maturity at least a trunk. You have to allow the trunk size minimum 10 inch, 12 inch. Then you can inject it. Otherwise, say, it is not. Even if we inject in where it is possible, that means that trunk size must be say, must be in shape that we can bore it. If it is very small, what will happen? Whenever we are going to bore it, then it will break. It will break. That is
Okay,
why we
sir.
have to allow at least 6 7 years uh, to develop.
So there is one question from Dr. Biplab Ghosh, and it says, "Is there any antioxidant component present in agar plant?"
Yes, in agarwood oil also shows some antioxidant activity. Number of papers are there. I have seen antioxidant activity is there. This agarwood oil.
So we would be most grateful, sir, if you would share the slide with us. Then we would have a better idea about the whole process. You know, the several aspects of cultivation as well as production, and the
If,
implementation of your ideas into practice, sir. So please do
yeah,
us the favor of sharing it, sir.
here some of the slide I can share. These are also available, but few
Okay, sir.
slides are there. I cannot share because I have signed in an agreement with DBT, so I have to.
Okay, sir.
give them to them.
Sir,
Already I have given so this is not
and sir,
my single
yes,
work.
yes, sir.
Seven researchers are working from the various corner of this country in this work. Already the project is completed. Now I shall submit it to them.
And sir, there was a specific request from the principal chief conservator, additional chief conservator of forests, asking for your slides so that it can be used for implementation purpose of the government policies. So therefore, kindly keep this also in mind. If you feel that it should be shared for the benefit of the public, then definitely we would like to request you to share the information with Praveen sir.
Okay.
So there is a question by Chiranjit Shutradhar. He says, "So why cultivation of these trees weren't made free like other cultivated trees? More because it is an expensive tree, which is also economically, which will strengthen the state economy, and therefore it should be made free. So has there been any initiative taken by the government regarding this?"
Initiative taken by the government.
Regarding making this cultivation, the cultivation of agar wood free for the public, or
That's
promotion
a
of yes.
That's I do not know actually the government what they are in newspaper I have seen some news. So in that in this purpose how can I tell? Just if anything related with the research based or related with the research activity, then only I can tell.
Okay, so we have one last question from Aditya Karmakar, a student of NSM. He has asked recently, he has planted 2 mahogany saplings at his home, and now he wants to know, can he plant the agar sapling close to that? Is it
The what?
feasible? Can he plant the agar sapling?
Planted two mahogany sapling
Yes.
at my home. Can I plant the agar sapling close to that? No problem. That means close to that means if he plant it just at the base, then how it will grow. But mahogany or acularia and acularia can grow with number of species. If jackfruit is there, so some other plant, mango plant is there, no problem. That should grow. Even in my second slide, some betel nut is there in this plantation, betel nut. It grows with betel nut. In Dharmanagar side, what happened? Their betel nut is again there. It is a popular species to them. So they planted betel nut and acularia at a time in the same place.
Patriot madam,
Yes. Yes.
আমাকে could you give me 2 minutes for I want
Yes,
to
sir.
say something to my students.
Yes, sir.
হ্যাঁ
Kindly continue, sir.
স্টুডেন্ট আমি স্টুডেন্টদের উদ্দেশ্যে বলছি, দেখো অ্যাকচুয়ালি প্রথমে আমি যখন ছোটবেলায় ছিলাম তখন রাবার কিন্তু তখন কাল্টিভেশন শুরু হয়নি। পরবর্তী সময় রাবার নিয়ে আমরা অনেক গল্প শুনেছি। প্র্যাকটিক্যালি আমার বাড়িতে রাবার কাল্টিভেশন হয়েছে এবং আমার পড়াশোনা পুরাটাই কিন্তু মানে কলেজ লেভেল থেকে অন দা বেসিস অফ রাবার। আমার বাড়িতে কেউ মানে চাকরি করার লোক ছিল না, নাম্বার 1। নাম্বার 2, আগর নিয়ে আমি অনেক গল্প শুনেছি। যেমন প্রথমে আমি জানতাম আগর, প্রথমে হয়তো আমি মানে অনেকে বলতো যে আগরের দাম আছে, সবকিছু জানতাম, কিন্তু অনেকে বলতো, আমার কাছে মনে হচ্ছে কেউ বলতো যে কাঁঠালের মতো বড় বড় ধরে, কেউ বলতো গাছ ফেটে কলার থুর যেমন ওই রকম বেরিয়ে আসে, ঠিক আছে। পরে এটা রিয়েল ট্রুথ আমি জানতে পেরেছি, যেমন ডক্টর দেবনাথ থেকেও মানে অনেক কিছু জেনেছি আমি, ঠিক আছে। তাহলে ওইটার যে ইকোনমি, ওইটাও কিন্তু একটা সময় মানে এই যে আগরের যে আজকে প্রবীণ স্যার বললেন, এগুলি আস্তে আস্তে উঠে যাচ্ছে রেস্ট্রিকশন গুলি। তাহলে এইটা আমাদের ফিউচার মানে হবে। আমি প্র্যাকটিক্যালি আমার বাড়িতে 2টা প্ল্যান্ট এরকম আগার রুট লাগিয়েছি এবং এগুলিকেও আর্টিফিশিয়ালি ইনিকুলেশন করা হয়েছে। আমি জানি না বিফোর 3 ইয়ার্স, এখন হয়তো টেস্ট করলে কিছুটা হতেও পারে। ওই রকম আমরা আরো গল্প শুনেছি, যেমন ধরো এই যে তক্ষক, এগুলিরও নাকি হিউজ মানে ডিমান্ড বাইরে। তো ইন ফিউচার মানে এগুলিও যে মানে ইকোনমিক ডেভেলপমেন্টের জন্য ত্রিপুরায় আসবে না, এটা আমরা জানি না। এখন এই যে তক্ষকের গল্পটা আমি এখনো জানি না। আমি জাস্ট মানে এমন আমার একটা লেবার থেকে আমি শুনেছি যে স্যার এমন তক্ষক একটা মানে 2 লাখ 3 লাখ টাকা বিক্রি হয়। আসলে এটারও পিছনেও কোনো না কোনো সায়েন্স আছে। তো স্টুডেন্ট যারা আছো মানে বা যারা মানে সায়েন্টিফিক কমিউনিটির লোক আছে, তারা নিশ্চয়ই টুমুরো এটাও ফাইন্ড আউট করবে। ইন ফিউচার এটাও হতে পারে যে একটা ভালো ইনকামের সোর্স, ঠিক আছে। এই তাহলে এই যে আমাদের আগরের লেকচারটা, এটা থেকে তোমরা এই জিনিসগুলি শিখো যে মানে ইন ফিউচার রেস্ট্রিকশন গুলি উঠে যাবে। আমরা যদি বাড়িতে আম গাছ কাঁঠাল গাছ যেমন লাগালাম, তার পাশে 2টা আগর গাছও লাগিয়ে দিলাম, হয়তো দেখা যাবে ওইটা আগে ছিল আনসার্টেনিটি, যে ন্যাচারাল ইনফেকশন এটা ভেরি ডিফিকাল্ট, এটা কখন হবে টোটালি আনসার্টেন, কিন্তু এখন তো আর্টিফিশিয়াল ইনোকুলেশন পাওয়া যাচ্ছে, আমরা ইনফেক্ট করে করে নিতে পারি। এই হচ্ছে তোমাদের মানে তোমরা এই জিনিসগুলি একটু দেখবে, এটাই আমার মেসেজ ছিল আর কি। থ্যাংক ইউ ম্যাডাম।
Thank you very much, sir, for giving additional information regarding Agar plantation or any other such plantations that the students can, use in order to make themselves self-sufficient. And we are very much thankful to our resource person today, Dr. Bimal Debnath, faculty of Tripura University. And now, if there are no more questions from any of our participants or from any of our resource persons or organizing committee, I would like to hand over this time to Dr. Partha Sharathi Debrai, the organizing secretary of this webinar on Agar wood as a potential bioresource for livelihood generation and climate change mitigation in Northeast India, to deliver the vote of thanks.
শুভ সন্ধ্যা, নমস্কার। Thank you, Patriot. A lovely and warm evening to our most honorable guest, resource person, principals, are organizing committee members, respected faculties, participants, and beloved students. It's a matter of pride for me that I am, as an organizing secretary, proposing a vote of thanks for this virtual meeting organized by the Department of Human Physiology and Botany of Netaji Subhas Mahavidyalaya. Thanks to all the people who helped us, either directly or indirectly, to make the virtual meeting successful. Thanks to all the participants for attending the meeting and more. I want to thank the Honorable Chief Guest, Sri Praveen Agarwal, IFS, Additional Principal, for his kind invitation and busy schedule to grace this meeting. On behalf of my college and the entire organizing committee members, I am very hearty gratitude to the speakers of today's virtual talk, Dr. Bimal Debnath, Assistant Professor, Department of Forestry and Biodiversity, for his valuable and lucid presentation of the topic on agarwood, a potential bioresource for livelihood generation and climate change mitigation in Northeast India. I think all participants will be benefited with his valuable lecture. I hope from his discussion, our students, participants got opportunity of the bioresource which may change their future ambition towards a new dimension. I hope Sir will share his presentation to us so that we will circulate to the students. I must acknowledge our greatest appreciation to our principal in charge, Dr. Sudhan Debnath, for his constant initiative and support. We have been organized this virtual meeting. I must thank to HOD of Department of Botany. She is also organizing secretary of this meeting. I must say, without her support, this meeting would not be possible. I must acknowledge to the IQC convener and other IQC committee members for giving valuable ideas to make a plan for the virtual talks. I also want to thank all the faculty members of the college for their cooperation in this virtual meeting. We are fortunate to thank the dedicated and well-motivated team of our college who are aware of their tasks and are very concerned about the results. Lastly, but most sincerely, I will give a big thanks to the Patriot Burma, Assistant Professor and HOD in charge, Department of English, for his for her efforts by wonderful communication and IT skill as an anchor of today's program. I mention my sincere sense of gratitude to her. There is much bearing on the vote of thanks to express my emotions that correctly feeds the atmosphere. So I show appreciation to the people who have been the strength of this meeting. Once again, I would like to state that we are glad to all the speakers here over the virtual talks. Once again, I will thank to all participants. We thank you for being with us. It has remained a pronounced pleasure. Thank you very much. With the permission of our principal in charge, Dr. Sudhan Devnath, I conclude today's virtual talk. Thank you. Thank you very much.
পার্থবাবুরে
Good night.
অনেক থ্যাঙ্কস। ঠিক আছে।